Okay, we all know that the mitochondria is the powerhouse of the cell, although if we're being grammatically picky about it, the mitochondrion is the powerhouse of the cell. I digress. Mitochondria generate ATP in a process called cellular respiration, and cells use this ATP as energy to do stuff. So, here's cellular respiration and ATP synthesis, explained as fast and as thoroughly as possible. ATP synthesis is, at its core, just a combustion reaction involving glucose. Our cells just break this reaction up into multiple steps so that we actually get useful energy out of it and not just a bunch of released heat. We'll start our journey in the cytoplasm of the cell, the site of glycolysis. Glucose gets turned into glucose 6-phosphate in a reaction helped along by an enzyme called hexokinase. This reaction actually uses up one ATP molecule. In the same way that you gotta use money to make money, you gotta use energy to make energy in this energy investment phase of glycolysis. A side note on enzymes, they're just special proteins that make it easier for reactions to happen by lowering activation energies. You'll see them everywhere in cellular respiration. They all end in A's, so if you hear that, know that it's an enzyme. With the help of phosphoglucose isomerase, glucose 6-phosphate turns into fructose 6-phosphate, which turns into fructose 1,6-bisphosphate with the help of phosphofructokinase. Say that 10 times fast. The creation of fructose 1,6-bisphosphate also consumes one ATP molecule. From here, fructose 1,6-bisphosphate splits in two, turning into dihydroxyacetone phosphate, abbreviated DHAP, and glyceraldehyde 3-phosphate with the help of fructose bisphosphate aldolase. Only glyceraldehyde 3-phosphate can proceed to the next step, so before progressing, DHAP needs to be converted into glyceraldehyde 3-phosphate with the help of another enzyme called triose phosphate isomerase. The first half of glycolysis is done. Now for the second half. We'll follow the journey of one molecule of glyceraldehyde 3-phosphate, keeping in mind though that one glucose turns into two glyceraldehyde 3-phosphates. From here, glyceraldehyde 3-phosphate is turned into 1,3-bisphosphoglycerate with the help of glyceraldehyde 3-phosphate dehydrogenase. This reaction generates a molecule of NADH. More on this molecule in a bit. 1,3-bisphosphoglycerate is then turned into 3-phosphoglycerate with the help of phosphoglycerate kinase. This reaction produces one molecule of ATP. With the help of phosphoglycerate mutase, 3-phosphoglycerate is turned into 2-phosphoglycerate, which with the help of analase is turned into phosphenyl pyruvate, which with the help of pyruvate kinase is turned into pyruvate. During this last step, another molecule of ATP is produced. This takes us to the end of glycolysis. Note that this second half of glycolysis occurs twice for every one glucose, so we've gained 4 ATPs, 2 NADHs, and 2 pyruvates from the second half of glycolysis. To summarize what happened in glycolysis, we lost 2 molecules of ATP initially, but gained 4 in the second half for a net gain of 2 ATP. Our glucose is turned into 2 pyruvates, and we also have 2 NADHs. We're going to create a running tally of these products as we move through cellular respiration. What's next? Pyruvate oxidation, which takes place in the matrix or the innermost part of the mitochondria. A carboxyl group is brutally ripped away from pyruvate, releasing carbon dioxide and leaving behind a 2-carbon molecule. This 2-carbon molecule, which is called an acetyl group, loses an electron, and another molecule of NADH is formed in the process. The acetyl group is then attached to coenzyme A to form acetyl-CoA. At the end of 2 rounds of pyruvate oxidation, because remember, 1 glucose begets 2 pyruvates, we've created 2 carbon dioxides and 2 NADHs. Our acetyl-CoA then enters the citric acid cycle, also known as the Krebs cycle, which also takes place in the mitochondrial matrix. Here, 8 more reactions happen. Acetyl-CoA combines with oxaloacetate and is turned into citrate, also known as citric acid, hence the name of the cycle, with the help of citrate synthase. Citrate turns into isocitrate with the help of aconitase. Isocitrate turns into alpha-ketoglutarate with the help of isocitrate dehydrogenase. In this process, another carbon dioxide and NADH are produced. Then, alpha-ketoglutarate turns into succinyl-CoA with the help of alpha-ketoglutarate dehydrogenase. Similar to the previous reaction, another carbon dioxide and NADH are produced. Succinyl-CoA is then turned into succinate with the help of succinyl-CoA synthetase. This reaction produces an ATP. Succinate is then turned into fumarate with the help of succinate dehydrogenase. This reaction produces a molecule of FADH2. More on this molecule in a little bit. With the help of fumarase, fumarate combines with water to form malate. In the last step, malate is converted back to oxaloacetate with the help of malate dehydrogenase, generating an NADH in the process. This oxaloacetate can then combine with another acetyl-CoA, hence why this is a cycle. To summarize, in a single cycle, we've created 3 NADHs, 1 FADH2, 1 ATP, and 2 carbon dioxides. For every 1 glucose, the cycle runs twice because 1 glucose molecule begets 2 pyruvates begets 2 acetyl-CoAs. So 1 glucose molecule generates 6 NADHs, 2 FADH2s, 2 ATPs, and 4 carbon dioxides in the citric acid cycle. Okay, on to the last step of ATP synthesis, oxidative phosphorylation. So remember those NADHs and FADH2s we were talking about? They're really important. They're actually responsible for creating most of the ATP. They're electron carriers, and their job is to transfer their electrons to the electron transport chain, or ETC, a series of proteins and molecules found on the inner membrane of the mitochondria. At the end of the ETC lies oxygen, the final electron acceptor. Oxygen combines with these electrons and some hydrogen ions to make water. The electrons from the electron carriers go wee as they slide down the ETC. The energy released by their shouts of joy as they slide down compels hydrogen ions to move past the inner membrane and into the intermembrane space. But then, these hydrogen ions realize they actually really miss the mitochondrial matrix, so they rush back in, pushing past ATP synthase in the process. ATP synthase gets mad and spins out of control, thereby generating approximately 28 ATP in the process. Okay, so maybe electrons don't scream, hydrogen ions don't miss the mitochondrial matrix, and ATP synthase doesn't get mad. But the analogy isn't terrible. Electrons do release energy as they move down the ETC, and this energy is used to pump hydrogen ions across the inner mitochondrial membrane. This creates an electrochemical gradient. Things always want to move down their electrochemical gradient, so these hydrogen ions want to move back into the matrix. To do this though, they must cross the inner membrane again. The only way to do this is to move through ATP synthase, a protein that spans the entire membrane. ATP synthase is essentially like a turbine in a hydroelectric plant. Instead of a flow of water though, it's a flow of hydrogen ions that spin the turbine, and instead of powering your toaster, the energy generated by this flow is used to make ATP. This process, in which energy from a hydrogen ion gradient is used to make ATP, is called chemiosmosis, and it's how the bulk of the ATP is generated. This is how our cells make energy. That's ATP synthesis, explained as fast as possible.